everyone. I'm Anjali Krishna, PhD scholar at Amrita School of Biotechnology. My topic is inferring variants of unknown significance in rare disease genetics. It's an India-centric study, and also my supervisor, Dr. Prashantan Surajala, and my co-supervisor, Dr. Renuka Surajala. And also talking about the rare disease, in our world, half of them are already affected with the various kind of rare disorders. If you're taking the India, because we are mainly targeting on the Indian population, so 140 crores people are living in India. Already 7.8% of the populations are tagged as rare, like the other countries also. So compared to the common disorders, rare disorder studies are really less. And also it's really a challenging phase. So we are mainly targeting on the rare diseases. And it could be monogenic, heterozygous, and migratory frequency. It should be less than 0.01. Many of the uncharacterized mutation will be there, and also largely we are targeting on uncertain and unknown significance, and it could be de novo mutation or rare mutation. And these steps are simply giving here next genomic sequencing mutations, and we are mainly targeting on the variant of unknown significance. And as per the ACMG guidelines, the classification of variants are classified as 5 categories: benign, likely benign, pathogenic, likely pathogenic, and uncertain significance, and the validation. So coming to the main disease, like we are mainly targeting on the CPC. CPC is a condition of a shortened pouch-like delayed colon, and mainly associated with the anorexia malformation. The pathogenesis and structural, it doesn't even understood, but we are suspecting like environmental factors and family inheritance may contribute to the pathology, and we are mainly targeting CPC because in India, the review show like a 92.2% of cases were here. And also, I have a hypothesis to check whether or not genetic variation is seen in non-coding RNAs, and also find the unique role in their pathogenesis. And also coming to the methodology, review of literature, and first we done the review of literature, then we are collecting the data set from the various sources, NCB and Indigenome, then data access and retrieval, then we are classifying the 3 categories: India, World, Asian, and also again we are going through the filter and annotation through claim 1 and batch analysis. We are categories 3 different categories, like likely pathogenic, uncertain significant, pathogenic. Then we are done the comparative analysis, taking the 2 sources, and we find out the total mutations, and there we jot out the 2 specific genes depending on the conditions and a role in their pathogenesis. And these are the result. Here mainly we are targeting on CAP4 gene, and also hereditary cancer predisposing syndrome based on the condition. If you're taking the significance also, it's causing pathogenicity and uncertain significance, and 2 of the genes. Coming to the conclusion part, the TCGA is a large-scale research project that has been sequenced and analyzed the genome of thousands of cancer samples, and this data shown the HNPCC is associated with the mutation in DNA mismatch repair genes, where responsible for the repair error in DNA replication. These mutation can lead the development of the colorectal cancer, and also 2018 and 2020, the CAP4 gene studies were conducted, and one of the studies is saying CAP4 gene was expressed in the development of the colon of mice, and the mutation of this gene could lead the defection of the colon development. And 2020 study was saying the CAP4 associated the gene associated with an increased risk of the colon cancer, and the study suggests that the CAP4 gene may play a very important role in the development of the colon cancer. Then the next one, conclusion: identifying pathogenic or variant of unknown significant SNP across the following rare disease phenotypes in all over India, and developing the causal relationship, establishing the causal relationship of non-coding regions, and identifying the genetic variation in them in some selected rare disease, and the anorexia malformation of India. And also we identified the candidate genes hereditary mutation like CAP4 associated with the colon cancer, and we are mainly targeted on the variant of unknown significance. These are the references. Thank you.